Welcome to section 9.3, Transcription. In this section, we'll explore transcription, the first major step in the central dogma of molecular biology. Transcription is the process of making an RNA copy from a DNA template. It's essential because DNA never leaves the nucleus in eukaryotes, but the cell still needs a way to use that information to build proteins. Transcription provides that messenger. By the end of this section, you should be able to explain the central dogma, the overall flow of genetic information in the cell. You'll also be able to describe the three stages of transcription: initiation, elongation, and termination. And finally, you'll be able to explain the processing steps that eukaryotic mRNA undergoes before it's ready to be translated into protein. The central dogma describes the flow of information in cells. DNA is transcribed into RNA, and RNA is translated into protein. This framework helps us understand how the instructions in DNA ultimately create the traits we observe. Notice that while the human genome has about 20 to 25,000 genes, the number of RNA transcripts and proteins is much larger. That's because many genes can be spliced in different ways, and proteins can be modified after they're made. This complexity is part of what makes biology so dynamic. This slide illustrates how a gene sequence is transcribed into mRNA. M stands for messenger. Only one strand of the DNA is actually copied during transcription, the template strand. Students often ask, how do we know which strand is used? And the answer is, it depends on the orientation of the promoter. The promoter signals RNA polymerase where to bind and which direction to read, so the template strand is chosen automatically. This ensures the mRNA matches the coding sequence of the gene, except it uses uracil in place of thymine. Transcription has three stages. In initiation, RNA polymerase binds to the promoter region of DNA and unwinds the helix. In elongation, the polymerase travels along the template strand, building an RNA chain by adding nucleotides one at a time. DNA unwinds in front of the enzyme and rewinds behind it, like a moving window. Finally, in termination, the polymerase reaches a signal in the DNA that tells it to stop. The mRNA is released, ready for processing. It's important to note that transcription is similar to DNA replication, but only one strand is copied, and the product is RNA instead of DNA. In eukaryotes, the first transcript produced is called pre-mRNA. Before it can be translated, it must be processed. Several things happen. A protective cap is added to the 5' end, a poly A tail is added to the 3' end, and stabilizing proteins help prevent degradation. Introns, or non-coding regions, are cut out, while exons, the coding regions, are joined together in a process called splicing. This editing ensures that mRNA contains the correct instructions for making a protein. An interesting twist is that splicing can occur in different patterns, producing different proteins from the same gene. This is called alternative splicing and is one way a limited number of genes can create so much complexity. The central dogma describes the flow of information from DNA to RNA to protein. Transcription is the process that creates RNA copies from DNA, carried out in three stages: initiation, elongation, and termination. In eukaryotes, the RNA must then be processed. It's stabilized with a cap and a tail, and introns are removed through splicing. These steps ensure the final mRNA is stable, accurate, and ready for translation in the cytoplasm. Together, these processes ensure that the instructions in DNA can be faithfully expressed as proteins, the molecules that do most of the work in cells.